All right, good afternoon, everybody. So welcome to today's physics colloquium, and it's my great pleasure to introduce Nikta Fakhri from MIT. So she's in the Department of Physics there. By way of background, Nikta's from Iran, where she went to the Sharif University and studied chemical and petroleum engineering, which is pretty interesting, and it's worthwhile to talk to her about those kinds of choices. From there, she went to Rice University, where she got her PhD in chemical engineering, and then went to the University of Göttingen, where she did a postdoc with Christoph Schmidt. And then she came to MIT, where she's been for roughly the last 5 years in the Department of Physics. And what I would say is that Nikta is working at a really interesting area at the confluence of biology and biophysics and non-equilibrium physics. And probably many of you know that there's a lot of excitement these days in the sense that people are trying to articulate new ideas about non-equilibrium systems, leading to things like fluctuation theorems. There's various predictions, and it turns out that some of them are most conveniently tested in living organisms. In fact, I think that there's an interesting argument that could be made that biology is offering a very nice gift to physics in the form of cool systems for thinking about non-equilibrium. Nikta has all sorts of really interesting external interests, and the one that I find probably most appealing is she likes to climb tall mountains, and she has recently done Mount Kilimanjaro and is getting ready shortly to do Mount Aconcagua, which some of you will know is the highest mountain in South America. It's actually quite high and cold and stuff. At any rate, it's great to have you here, and look forward to your talk.
Okay. Thank you, Rob, for the introduction and also invitation. I've had a wonderful day today, and I'm looking forward to telling you about some of the work that we've been excited about over the past few years. And I thought it was only appropriate to show basically Feynman ratchet here. And this is kind of like the artist's depiction of the Feynman ratchet. And what you see here on the surface is like a sheet of cells. And what I'm going to show you is how we have been applying ideas from stochastic thermodynamics and non-equilibrium physics to try to uncover mechanisms of activity in biological systems. And I will start by first motivating why we're really excited about biological systems and what they can offer. And one of the distinctive features of living systems is their faculty to move, to divide, to change shape, and to define their own morphology. And to highlight this kind of varied range of functions, I'm going to focus on my favorite model system that we use in our lab, starfish oocyte. We like to call it the hydrogen atom of development because, in fact, it's actually the first experimental system that was used to study development. And you can obviously imagine that from a physicist's point of view, these systems are highly fascinating, but at the same time they're quite frightening, simply because of the number of variables that would be necessary to describe such varied range of behavior. And so, for instance, here, this is actually the starfish that we get from in the coast in Santa Monica. So we have a diver who goes and dives and sends us via FedEx shipment of starfish, which you can keep alive in a physics lab. And then you can see that the starfish starts from a single cell, oocyte, which is about 180 micron. And this cell has to undergo multiple cell divisions, whereby a large cell like this has to find its mid-plane, and then undergoing multiple rounds of cell division, morphological changes, and finally developing into this small larva, which is swimming, and then finally developing into the full organism. And for the sake of maybe a little bit simplicity, I'm going to focus on at a single cell level. And cells are elementary building blocks of living systems. They're extraordinarily dynamic, and they can perform a varied range of functions. And to perform such a varied range of functions, they rely on ordered structures and intricately controlled collective dynamics. A canonical example of such structure is cell actomizing cortex. And I'm going to spend a little bit of time to introduce what this system is, and this is all the biology that we will need for the rest of this talk. So the actomyosin cortex is a quasi-two-dimensional network of a type of protein which is filamentous and known as F-actin, which polymerizes from the cell membrane and forms a very thin layer underneath the cell membrane. So this is basically, it's underneath the cell membrane, and here you can see a faint color of red or magenta. And this, at first glance, it looks like a physical gel, which is percolating because you have cross-linking proteins that basically cross-link this gel, and they have some certain on and off rate. But on top of that, you also have another type of protein known as molecular motors. These are energy transducing proteins. In this particular case, myosin is the motor protein of interest, which they basically bind to our actin network, and they locally generate stress. So just think about they stochastically bind to your cortex. They're force dipoles. They generate some kind of stress, and then they release from the network. And this happens stochastically throughout this network. So you have stochastic stress fluctuations happening in the system like this. And of course, because these are energy transducing proteins, it means that they're constantly hydrolyzing ATP, or the fuel, and then generating mechanical stresses and work. And because of these interesting features, these gels exhibit, these are active gels, and they exhibit really interesting properties, which you don't really see them in equilibrium counterparts. And I want to highlight a few of these really important features of active systems like this. First of all, we have an inverse energy cascading happening in the system. So you have energy injection at the molecular scale, and then this basically cascades to much larger scale. So you have motion and stress generation on much larger scale. And this is kind of an example of a class of non-equilibrium systems known as active matter, where, again, the drive doesn't happen at the boundary. The drive happens inside the system at the molecular scale and then gets propagated at much larger scale. Because of this continuous injection of energy at molecular scale, you have spontaneous self-organization of these structures, which are actually non-equilibrium steady states. Again, this is different from classical non-equilibrium systems, where what you are really interested in is pushing a system away from the equilibrium and seeing how it relaxes back to equilibrium. This actually is, these structures are non-equilibrium steady states that persist for time scales that are much larger than molecular time scales. And examples of this length and time scales are, for instance, in the case of actomyosin network, this network is responsible for the mechanical integrity of the cell. So you can imagine you have this thin sheet of actomyosin network underneath the cell, and that's how the cell keeps its shape. And this mechanical integrity or stiffness happens on the order of hours, whereas molecular turnover rates are on the order of minutes. As a matter of fact, during this video, basically the whole network polymerized and repolymerized, so none of the proteins were the same as when I started the video. I think this is an amazing kind of feature, because imagine you are driving across a bridge, and as you are driving, the bridge is being assembled and reassembled. And you also have emergence of new time scales, length scales in the system, and in particular, for instance, in the case of cell division happens on the orders of microns, whereas the molecular scales are the orders of nanometers. And again, these proteins, they have to self-organize into these structures, and so that in a way they can find where the middle of the cell is, so that they can exert force and basically divide the two cells very precisely into two daughter cells. So I think this features, and hopefully now everyone knows what the system is, and these features highlights the fact that there is a lot of new physics that can be done in biological systems. And I really like this quote by Stan Ulam, where basically biology is a playground for doing new types of physics, and where you can really push the limits of understanding systems that are far away from equilibrium. So for the next remaining of the talk, I'm going to focus first on one particular question that we've been very excited about in my group, and then towards the end I'm going to give you a flavor of other type of questions that we have been trying to answer, and then how somehow these two type of questions merge, and what is the big picture here. And today, in particular, I would like to see if we can use ideas from non-equilibrium thermodynamics to try coming up with frameworks which allows us to not only detect the non-equilibrium activity in this highly fluctuating systems where you have both thermal fluctuations and active fluctuations, and also come up with ways of quantifying distance from equilibrium. Again, these are true non-equilibrium steady states. But then also quantifying the amount of dissipation. And the big picture here is that can we actually come up with an energetic landscape for the cell and map how a cell is budgeting its energy for different type of processes. And I'm going to show you two such frameworks. First, I'm going to show how we can use a broken detailed balance at mesoscopic scale to quantify if a process that we're observing is equilibrium or non-equilibrium. So it gives us a possibility to go away from standard fluctuation dissipation theorem and just by observation say something about the system. And then finally, I'm going to focus on another framework that I'm really excited about. And the idea is that can we come up with ways of measuring this arrow of time? And I'm going to show you how we can come up with entropy-like terms, which allows us to not only quantify this arrow of time, but also get access to the underlying time scales of the system in a completely model-independent way. So first, let's focus on detailed balance. And I'm going to just give you a very quick refresher of principle of detailed balance. And of course, this principle was used by Boltzmann to prove his H theorem. And the idea is that if you have, when a system reaches steady state, so this is a system of three states, when a system reaches steady states, its properties become stationary in time, which means that if you look at each state, the transition in and out of any microscopic state of the system is balanced. However, equilibrium puts another constraint on the system known as detailed balance, which means that if you look at any pairwise states, microstates in your system, the transition rates between these pairwise states should also be balanced. What it means is that currents and cycles in the phase space of microscopic states of the system are forbidden. Now, this is obviously what's at the heart of thermodynamic equilibrium. However, biological systems operate far from equilibrium, and violation of detailed balance is at the heart of their dynamics. And this is actually really nicely demonstrated by this video here. So this is a single molecular motor, one of this energy transducing proteins. It's a different system. It's called kinesin, and this is another filamentous protein known as microtubule. But the idea is that this conformational states or microstates of this protein gets coupled to ATP hydrolysis, so some of the states become more likely, and then what happens is that this motor protein on average walks in a particular direction. Right? So this is really at the heart of biological systems. So at molecular scale, these systems break detailed balance. But now the question is when we obviously, this breaking of detailed balance is important because in contrast to thermal noise, non-thermal noise can generate spontaneous motion. And again, this is well illustrated by Feynman's ratchet thought experiment. And a perpetual motion does not happen at equilibrium, but in non-equilibrium, or when, for instance, you put your paddle and pawl at two different temperatures, you can actually generate spontaneous motion. But you can see that ideas like this was also applied to molecular motors, which are isothermal systems. However, the energy comes from this ATP hydrolysis. So now what we want to basically understand is, obviously non-equilibrium is important for functions of biological system. It can boost intracellular transport. It can increase the fidelity of transcription, chemotaxis, and also accuracy of a number of different processes. And to understand cell function, then it would be important to determine whether particular cellular processes are result from a non-equilibrium activity. But this can be, of course, challenging. For instance, if I show you videos, and it doesn't matter what the systems are, but when you look at a system like this, it's immediately clear that this is a system that has some kind of drive, right? So it has a particular beating or directed motion. But if I show you a system like this, so this is, and these are mesoscopic systems, so we're basically zooming out. We're going away from a function of a single protein, and you have thousands and thousands of protein here at play. With this type of motion here, it's not clear if it's really driven by thermal fluctuations. Obviously, this is a soft matter system. At this scale, thermal motion is pretty important. Or are these fluctuations results from some underlying non-equilibrium process? And more importantly, it has been shown that a detailed balance can re-emerge when you start coarse-graining the system. And somehow you can think of if you can find the scale beyond which detailed balance is restored, maybe you can then, by all means, the system behaves like equilibrium, and you can use equilibrium stat-mech to describe the macroscopic properties of the system. So how can we kind of try to implement this detailed balance violation at much larger and mesoscopic scales? So let's start by looking a stationary dynamic system that evolves on time in short time scales, either due to thermal and non-thermal fluctuations. And we experimentally, we only have access to a set of degrees of freedom in the system, which is denoted here by D. So this is a subset of degrees of freedom in the system, and these are configurational degrees of freedom. So in systems like this, the momental degrees of freedom relaxes very fast, and so configurational degrees of freedom are the most important one. And we don't really have access to the other the full subset, basically the other subset of degrees of freedom in the system. So what I'm going to do is I'm going to integrate those out. So because the system is stationary, you can describe its properties by a probability density of how likely it is to be in particular state, and you can write the continuity equation for the system. And of course, what's important is that at steady state, you have nabla.j 0, right? So now we're going to see if we can apply this to observational through the observations that we do in experimental system. And the idea is that let's look at, for simplicity, let's look at 2 degrees of configurational degrees of freedom that I can extract from experimental systems, so x1 and x2. What I have is a trajectory in this continuous configurational phase space. Now what I'm going to do is I'm going to coarse-grain this phase space. I'm going to choose some coarse-graining parameter and generate this mesoscopic states. So now I have my random walk is basically now a Markov type process. And what I'm going to do is I'm going to, for each of these mesoscopic states, I'm going to measure the probability density of being in a particular state by doing averaging. And I'm also going to count the transition rates between neighboring states. And what that allows me to do is to construct a flux. And the flux is here denoted by J. And basically the it's aligned by to the covariance matrix of J1 in one direction and J2 in the other direction. And then through bootstrapping to make sure, I mean, these are statistical type measurements, so we want to make sure that the currents are statistically significant. So via bootstrapping, we can actually find how significant these currents are. And for each of these J's that I will measure, I'll put an ellipse here, which is basically standard error of the mean. So let's see if we can first apply this to a simple toy model. And the question is that, again, we're interested in stochastic, highly stochastic systems, and this system is, again, in the same class of models of a Brownian ratchet. And what we have here is, so it's two overdamped feathered beads that are connected with a spring here. And what we're going to do is we are measuring the position of each of these beads. So these are the position trajectories you can see plotted here. And what we're going to do is we're going to change the temperature of these two beads and then see if, again, the trajectories are fully stochastic. But then the question is that in this fully stochastic system, is our phase space analysis, coarse-grained phase space analysis, will allow us to see cycles and loops. So, so first let's look at the scenario where the two temperatures are the same. And again, what we're going to do is, so we have some random trajectory of these two configurational variables, x1, the position of bead 1, and x2, the position of bead 2. And we do our coarse graining, and then we can see that the currents are not significant. So we don't see, actually, the currents, the flux is 0. Now let's take one of the beads here, this red one, and put it at a higher temperature. And if you do that, you immediately see that we can see a cycle in the phase space of all the states of the system. So this actually allowed us to be confident about the fact that even a fully stochastic system, we can try to implement this idea. And to test this, we are going to back to one of the experimental videos that I showed you. So let's see what exactly this video is. So what I have is a cell. So this part of the video is a cell. And this is the cell cortex that I talked about. So you can see that there's a lot of different organelles, and they're just fluctuating around. Well, what's really important here is I have this kind of organ here, which is called a cilia, and it's actually a mechanosensor. So this cilia can senses the flows, and what it does is it signals to some other proteins, or other proteins that are sitting on the membrane, so the membrane knows when it has to open or close up for ions to flow in and out. So again, if you look at the dynamics of this cilia, by all means, it looks like a thermal fluctuation. It's just a Brownian motion. So we're going to try to come up with two degrees of freedom where we can measure those experimentally and then do our phase space, coarse-grained phase space analysis. So the 2 degrees of freedom we decided to choose was, one is the average angle with respect to the vertical line to the cell surface. So this is, again, it's in this cartoon here. This is theta. And the other one is the average curvature of the cilia as it undergoes fluctuations. And let's look at the trace of these trajectories. So the angle in both the curvature as a function of time looks completely random and stochastic. So now I'm going to implement my phase space analysis. So we're going to generate a phase space of angle and curvature. Again, what we end up having a random walk in this space. And then we're going to do coarse graining. And then the color here is the probability of being in a particular state, which we did by doing, which we measured by doing averaging. And then we also looked at the transition rates between the neighboring states, and then constructed our J vector. And then what you can see is that on average, we have a clockwise cycle in the middle of the phase space here. So this is exciting because, and again, the ellipsis here tells us how significant, doing bootstrapping, how significant these currents are. So this is really exciting because in a completely data-driven way, so just by observation, you can actually quantify a process as being driven thermally or with some active drive is important. And to kind of, this is obviously a biological system where you can do perturbations to get rid of what's the source of the active drive. And for this particular system, we knew that what's driving these fluctuations are the base of the cilia is embedded in actomyosin cortex. And if you remember, we had this myosin motors that stochastically bind to the cortex. They generate some stochastic stress fluctuations. And that actually was driving the fluctuations of the cilia. And it basically manifests in this type of dynamics that we observe. So if we can do some kind of manipulation to get rid of myosin, which is the stress generator, you can see that our cycle goes away. So we basically recover a detailed balance in the system. So I just want to kind of wrap up this part of the talk. So what we show was it's possible to use very commonly available conformational degrees of freedom to identify non-equilibrium dynamics in highly stochastic steady-state systems. And this is intrinsically non-invasive. It's really only relies on observation, which has been developing quite a bit over the past few years. So you have more and more data because of the range of different microscopy techniques that are now available. But more importantly, it also doesn't rely on any other physical perturbation. So if you want to look at fluctuation dissipation theorem, for instance, which also is violated in non-equilibrium systems, you can observe the fluctuations. But for dissipation, you need to measure the response of the system. And that, again, is it's an invasive way. And it's very difficult to do in biology. But this actually, again, gives us a way of, just from observation, infer something about the dynamics in the system. It also doesn't rely on rare events. So all of these fluctuations are Gaussian. So it doesn't really rely on rare events where you have like the tails of the distribution becoming important. And we have also been working, we had a paper where we extensively studied the toy model to show that from these currents, you can actually look at the fluctuations of the currents to put bounds on the amount of dissipation or entropy production in the system. And I'm not going to go into details of that, but I'm happy to talk offline about it. But what's the drawback of this system? The drawback of this system is it's very much dependent on the choice of the observable. So if you happen to choose the wrong degrees of freedom, you can't basically say anything about the system if you don't see cycles. So, so again, this is it's powerful, but then it heavily relies on the choice of the degrees of freedom. It also relies on the type of coarse graining you do. So you can imagine that if your coarse graining level is fine, you don't have enough statistics to say something about the system. If you coarse grain too much, by all means, your cycles can go away, and so you really have to be you have to come up with ways of knowing what the correct coarse graining level is for the system. So is there another, so let's kind of like go back to the blackboard and think about what's at the heart of thermodynamic equilibrium and see if we can come up with another way of trying to not only kind of quantify, not only do a binary classification, but also say more about what happens in the system. And this is kind of this part I'm going to talk about how we've been using the idea of arrow of time to do that. And let's kind of like demonstrate this with a video that I really like to show. And again, if we go back to the first part of my talk, I talked about cell division and how it's important. So the video I'm going to show you is a video of a developing embryo. So this is the same embryo that I showed in the beginning. And what you see is you have a lot of cells. And as I'm going to play this video, you're going to see what happens. So let's take a minute to watch the video. So we started with about 64 cells. And then the video is playing. And if you are a biologist by now, you should be bothered. But in the physics audience, I always have to kind of take a little longer to say, okay, are you bothered by this video? So anyone thinks something is wrong about this video? Exactly. So this never happens in biology. So in biology, you don't get you get cell division. You don't get the merging of cells. So what it really highlights is irreversibility in non-equilibrium processes can be quantified in terms of, first of all, how much entropy the particular dynamics produces, but also the fact that we do have this notion of arrow of time in biological systems. And the fact that irreversibility encodes how much entropy particular processes produce is given by this really beautiful equation, which I'm going to walk you through. So imagine we have a stationary process. And for this stationary process, we're going to observe a sequence of events, which I'm going to call the forward sequence of events. And then I'm going to take the same sequence of events, and I'm going to reverse those. And you can show that if you look at the probability distribution of observing the forward sequence of events and the probability to observing the reverse sequence of events, you can compute an entropy-like term, which is here is known as Kullback-Leibler divergence. So basically, you're comparing how you're looking at how overlapping two distributions are, and in this case, we're looking at the forward and reverse distributions. And what's absolutely amazing is in this, for this equation is that you can show that in the limit of observing all the degrees of freedom in your system, this is the exact entropy production rate for your system. And I want to take a minute to, again, appreciate this equation, because I'm so fond of it. One side of the equation is a purely information theory metric. So all you do is you do an observation, and you compute these distributions, and you measure your KL divergence. But the other side of the equation is a fundamental physical quantity that you can measure in your system. Okay, so let's see if we can apply this idea. I mean, this idea has been was developed by Juan Parando, and it's obviously was theoretically it seems very powerful. But the question is that experimentally, how can you apply it, and how much data would you need? And indeed, would you? Of course, because we're not observing all the degrees of freedom, we are going to get a lower bound, which instead of calling entropy production rate, I'm going to call irreversibility. So a lower bound on the amount of dissipation that we observe. And it's kind of like easy to detect a video of like this is not symmetric under time reversal. So let's start with a much harder system. So I'm going to show you a video like this. So this is, again, I'm going to tell you what the system is in a minute. But this is just if you look at this video for some time, you can see that I have some spherical probe particles in some environment. And if I show you this video, immediately you can think of, oh, this is Brownian motion. What actually Brown was hoping to be active motion, but it was really just a thermal motion. But so I'm going to so this is a highly stochastic system, but I'm going to tell you what exactly this video is, and this is our actomizing cortex. So so this is kind of looking from the bottom here, and so I have actin cortex here, I have my myosin motors, and what I'm going to do is I'm going to introduce some probe particles, the spherical kind of objects here, and these spherical objects are the readout of the fluctuations in the system. So they're both kind of affected by thermal motion, which is always present, but they're also getting kicks from these stochastic fluctuations of the myosin activity. So again, if you notice in the middle of the video, I reversed this video, but you can see that it's really hard to see if there is any difference between the forward and reverse. So this was our challenge. And to kind of like to do that, we have all these probe particles. We can image them for very long times. And we end up, the data we get is this random trajectories. And because we need a stationary process, instead of looking position itself, we're going to look at position increment. And another reason to look at position increment is because it's also a readout of the stationary local force fluctuations. So your probe particles are embedded in a viscoelastic environment. So basically that is also, like, their dynamics is a readout, if you know all the material properties of the system, it's the exact force stress kind of pattern. But for now, let's say that it's just a readout of the stationary local force fluctuations. And one thing we're going to do, so to generate this position increments, we can introduce this theta here, which is a coarse graining parameter. And the idea is that I have all these position increment traces, and by increasing theta, what we are doing is we are kind of setting a scale in the system. So we are saying we don't care about fast fluctuations. We are going to average those out, and what's remaining is what we really care about. And this actually turns out to be an important kind of addition that we did that allowed us to get some really important information from a system like this. So all we need to know is we have all these traces, and larger lag time means more coarse-grained data. Okay? So now, just to give you an idea what we should expect to see from a system like this, let's look at the correlation function, the autocorrelation function of this position increments. And what you can see is that at time t0, we have a delta correlated noise, and then it becomes negatively correlated for fast time scales, and this is understandable because thermal noise acts on much faster time scales. So every time your particle gets a kick, it's in a locally elastic environment, it just goes back. So you have this negative dip in your autocorrelation. And it is only when you have your active time scale, which is the Meisen, the force dipole fluctuations kicks in, your particles diffuse away. And what's important is that there's a particular time scale of this Meisen activity, and we've shown that in another paper, which is basically it's at about 10 seconds. So if you look at the mean square displacement of one of these particles as a function of time, you can see that fast time scales, fluctuation dissipation theorem holds. So fluctuations are exactly what you would expect from the response function if FDT holds. And it's only at this intermediate time scales where the fluctuations are much larger than what you would calculate from the response function if FDD holds. And again, we understand this because of this additional force fluctuations of Meisen motor. So this 10-second time scale is important. Okay, let's now go back to the data and see if we can compute our KL divergence or irreversibility. So I'm not going to go into details, but we don't do this in real space. We do this in trajectory space. So you have this forward time series and reverse time series. And we can generate this sub-trajectories, and then you can construct an n-dimensional trajectory space where you can do histogramming to kind of construct the distributions, and then you can compute the irreversibility or KL divergence for the forward and reverse time series. Okay, and at the end, going through, I mean, this was kind of like a very huge achievement done by one of the undergraduate students actually in my group, which I'm going to tell you about in the last slide. But what's important is that we from each of these trajectories, we're going to get one number, which is this irreversibility. And I think the most exciting thing that came out of this analysis is now I'm going to plot irreversibility, this number, as a function of the coarse graining parameter lag time that I introduced. And what you can see is that it's a non-monotonic function of lag time. And it took a while for us, it took us some time to try to really understand what's happening in the system. But what we now know is that, as I mentioned before, fast time scales in this system is dominated by thermal noise. So when you measure an irreversibility, actually the process is reversible because it's generated from thermal noise. So this quantity is very small. And we're actually very proud of you had huge amounts of data to get good convergence to kind of get these numbers. And the error bars are smaller than the symbols here. But so this number is very small because it's really dominated by thermal noise. Long time scales, what you're doing is you're coarse-graining information out. So again, this is an information theory metric, so it's obvious that it starts going down. But what's really exciting is the fact that in between you have this peak. And if you go back and see that, look at the time scale of the peak, it's actually the time scale of your active drive. And what's exciting is that in a completely model-independent way, you actually get you will go and say, okay, there must be something important about this time scale, and then you can now start looking into mechanisms of what drives activity in a system like this. So just to show you that indeed, even from a simple model, you can also see how if you tune the time scale of activity, this knee actually drops. So we looked at, again, a simple model system of a particle experiencing active noise, thermal noise, and active noise. And then so it's kind of looking at Langevin dynamics. And so the active noise is it's an Ornstein-Uhlenbeck process. I'm happy to go to details if anyone is interested. But the point is that when we tune the active time scale, which is the ratio of this kappa to alpha, you see that indeed this kind of time scale shifts. So you indeed in a simple kind of toy model, you can also see this type of behavior. Now, one question that you might ask is, I told you that this is a lower bound number. But then what's kind of like the question you might ask is, how good of a lower bound is it? And if you give me different kind of configurations of the system where you can tune the amount of activity, with this lower bound, would I be able to distinguish between these type of systems? So this is a biological system, so we can actually tune the amount of active drive, and we can generate 3 distinct steady states. So you can, for the biologists in the audience, you can do ATP depletion, so you can get rid of the fuel of the cell to generate a low activity steady state. You can introduce more myosin, so you can express myosin to get a high activity steady state. And this is the wild type, the medium activity steady state that I have. And then now we're going to look at, so we had the peak, and then I'm going to look at the time scales way past that peak to make sure that what we are measuring is really the irreversibility. And what's, again, was really exciting was the fact that despite the fact that this is a lower bound number, we do have a monotonic relation between the amount of activity and the irreversibility number that we are getting from our system. And what's, again, another thing that was, to me, was absolutely amusing is we're looking at we're looking at biological isolates. So usually biologists look at systems that are genetically identical. These systems, we just get them from the coast, we bring them to the lab, and then we're looking at their kind of the biological processes. And then this difference is, that you can see here comes from cell-to-cell variability, which is, I think, I find that actually quite amusing. Okay, so I'm going to wrap up this part of my talk as well, which is basically we show that irreversibility, kind of the dependence of irreversibility on coarse graining, and the fact that we can get signatures of timescales important in the system doing this type of analysis. And the lower bound number is good enough that allows us to rank order the activity in the system. So now one can start thinking about, can we actually relate this number to the type of process that's happening in the cell, and this kind of moving more towards this idea of looking at an energetic landscape inside the cell. And one kind of so this was coarse graining in time. One can also think about coarse graining in space. So, so far, what we did was we introduced spherical probe particles inside the system. So these probe particles, they only have one length scale, which is the diameter of the probe. And we've been extending this technique to introduce filamentous probes, where we can now also look at the spatial extent of activity and how you can get energy cascading happening in the system. And I'm going to maybe briefly show how we do that. So for that, we use kind of my favorite nanoparticle from my graduate school, single-walled carbon nanotubes. In particular, we're interested in semiconductor nanotubes, which have really interesting optical properties. Just for very briefly, they have, so the semiconductor ones, they have band gaps, and you can excite the electrons in using visible range light, and then they emit in infrared region. And this is a bulk sample, so each of these points correspond to a particular chirality of carbon nanotube. Well, what's important for our application is the fact that they're very thin. They're only a nanometer diameter. We can tune the length. We can get carbon nanotubes from tens of nanometers to tens of microns. And one important thing is that they're pretty stiff, so they actually have the same stiffness as biopolymers, actin and microtubule that I introduced. And we've been using them in different contexts to probe biological systems. This is because they're super photostable, and because you are in the infrared region, you don't have background from biological species. But I want to actually focus on this video here. This is a very long nanotube, so it's about 15 micron. And we're basically using like the fluorescence, and we're imaging carbon nanotube. And what you don't see is what's in the background. And in the background, they actually have a gel. And what's absolutely amazing is the fact that you see this really distinct motion that's known as reptation, which was introduced by Dogène. So you can see that you have all these transverse fluctuations, and actually these transverse fluctuations encode information about the dynamics in the environment. So what you can do is you can take your carbon nanotubes and put them in a dead system and in an active system. Dead system is just the active gel. A live system is where we introduce the energy transducing proteins, myosin motors. And then kind of briefly just how we introduce length scale in the system is we are interested in transverse fluctuations, so we can decompose the shape of our carbon nanotubes into fundamental modes of a force-free, torque-free filament. And now so what you end up getting is traces of the amplitude of bending mode. So this is the first mode, and this is the amplitude of the first mode as a function of time, the second, third mode, and so on and so forth. So now you have an n-dimensional kind of space of amplitude of bending modes, and each of these modes encode information on a particular length scale. So you can imagine mode 1 is really looking at the length of your filament. Mode 2 is looking at a smaller and smaller length scale. And now we can apply the same idea of kind of irreversibility or breaking of detailed balance to time series like this. And I'm not going to go into details of that, but again, I'm happy to talk about that offline. So for the remaining of the talk, I just wanted to give you also a flavor of other type of questions that we are interested in, and then at the end maybe just give you a broad overview of where we think we're going with this with these ideas. And kind of one important feature, again, in biological systems, they're not equilibrium systems where you perturb and look at their relaxation to equilibrium. They're actually true non-equilibrium steady states. And these non-equilibrium states, you can think of them like the structure, which kind of encodes the function, is this non-equilibrium steady state. So one kind of important question would be, what are the kind of like important order parameters in this system? And obviously they undergo symmetry breaking. So what are the symmetries of the system? What are the broken symmetries of a system like this? And this has been something that we've been working on. And I'm going to focus on one particular kind of project, which is basically trying to look at the broken symmetries in a system like this. And I'm going to go back to the video of the cell division that initially I showed you, and I'm going to ask you guys to pay attention to one interesting feature here. So just before cell division happens, so all these colors that you see are proteins that are labeled with different fluorescent tags. And you see this flash of green kind of spirals that emerges. And you can see that the spiral starts far away, but then as the cell kind of undergoes cell division, you focus the spirals to the middle of the cell. And in fact, this is how the cell knows where its kind of center is. Because this kind of flash is a signaling protein known as Rho-GTP, which is it's upstream of actin and myosin activation. So this is kind of like the green dots here. So this Rho-GTP has two states. In the active state, it binds to the membrane. And when it binds to the membrane, and it's kind of like with a particular pattern, it activates actin. So it starts polymerizing actin. And it also activates myosin. So for the biologists in the audience, it phosphorylates myosin. So anywhere you have this protein binds, you have a network formation. And this is basically how the cell gets the signal that this is the middle and this is where the cell division should happen. And we actually have, so again, this is our protein of interest. It's upstream of actin and myosin activation. And all we need to know is it has two states. In the active state, it binds to the membrane. So this is the green kind of dots or shapes here. And then in the inactive form, it's in the bulk. So these patterns that you see are on the membrane, on a two-dimensional membrane. And what's kind of what we have shown is that you need another protein to activate this molecule from the inactive form to the active form so it can bind to the membrane. You can externally introduce more of these proteins in the system. And what happens is that this is an excitable system. You can, by overexpressing this protein here, you can actually push the system to a bistable region. So what happens is that each point on the membrane become bistable, so it undergoes limit cycle oscillations. So what you can then think of is basically we have a system of weakly coupled oscillators. And what we're going to do is we're going to look at kind of each point on the membrane. So I'm going to take one point on the membrane here, and then you can see that it's undergoing oscillations. And what we're going to do is we're going to get this oscillatory time trace. We're going to do a delay time embedding algorithm to find the phase of oscillation. And then at the end, what you end up with is this beautiful phase of the oscillation of this molecule, this protein on the membrane. And kind of like without going to details, it's been something that we have been quite excited about because you can show that these spirals, they actually, if you look at the phase, there is a point that the phase is not defined, and that actually is it's a topological defect of either +1 or -1, depending on the rotation. And now this has been quite exciting. This actually came about. I showed this video to my colleague Martin Zwarlein, and he's like, "Oh, this looks like quantum turbulence. Have you thought about thinking of a system in terms of this ideas and frameworks?" So we got really excited, and we kind of started looking at basically how these defects starts interacting in time. And this is kind of, you see that defects have not only have the dynamics happen you can see them in space, but then you have all these interesting dynamics that happens in time. And the first thing that we have showed is basically, again, you actually have a turbulent state where these defects are kind of behave just like quantum turbulence, where they have a particular, and this is the phase of the oscillation. You can look at the phase velocity and the vorticity. You can show that there's a certain vortex size that they start interacting, and then you can do some really interesting kind of physics with this. But again, I'm happy this was actually just accepted, and I'm happy to talk about that in offline. But one kind of other thing that we've been pushing this system now is if you look at again, this is the XY, and you have all these defects that, again, each point, so you have the +1 and -1. They're kind of like interacting. And what you can see is that you get loops, And when you can, the defects can exchange partners, so you can get really intricate loops, you can get knots, and then sometimes you can push the system to in a regime where you only get actually braiding happening. And this is something now we're trying to think about. Okay, we can maybe we can encode information in these loops and knots and kind of braiding that's happening. And this kind of, maybe I'll tell you about this another time. So I'm going to just kind of like quickly wrap up like how I think these two areas in my group can actually start interacting. So what I actually showed you is we have this kind of frameworks which basically tells us how much dissipation for particular processes is happening inside the cell. And then you have this really beautiful symmetry breaking processes that obviously happens in biology. And one idea that we really want to push is, can we use this irreversibility metric or kind of dissipation metric as an order parameter, which basically allows us to look at non-equilibrium phase transitions and symmetry breaking that happens in a system like this. But even kind of like from a broader perspective, systems like this are usually studied from a nonlinear dynamics point of view. But obviously, these are non-equilibrium systems. You have continuous, you need ATP for these patterns to get generated, for the symmetry breaking to happen, but also for the patterns to be maintained. And how much dissipation the cell requires, how much energy the cell has to expend to kind of maintain patterns like this, to generate patterns like this, is another question that we are looking forward to answer. And one kind of last thing is the fact that these patterns are signaling proteins, so they encode information. So we are actually looking at information transfer across the cell, and then one can start thinking about how much dissipation is required for information processing and transfer inside the cell. So with that, I'm going to wrap up my talk and get to the most important slide, which is basically all the amazing graduate students and collaborators that have been kind of helping us with this project. So this is all the graduate students in my group, and the work that I described today is mostly work of Zerhan Tan and Alex Bakanu. And the arrow of time irreversibility is the work of an undergraduate student in my group, Garrett Watson, who really joined over the summer, and I could pitch this idea to him, and he kind of really went after it. And we've been very fortunate to, at the same time that I joined MIT, we had two other postdocs that joined MIT. These are independent physics of living systems fellows, Jordan Horowitz and Todd Gingrich. And that actually, so they both theoretical physicists who are interested in stochastic thermodynamics, and we could really kind of come up with interesting ideas, and we hope to continue working along those lines. I have a number of collaborators both at MIT and across globe. In particular, Erwin Fry's group, where we have been looking at kind of what are the minimum kind of motifs that you need for this signaling, kind of the signaling Rho actin myosin cycle that I described. Jorn Dunkel's group, Jorn is at math department, and ideas of topology actually really started when Jorn and I was at a conference with Gil, and he talked about topology, and then Jorn and I was like, oh, we should do something with topology, and that really came about. And also Kinneret Karen's group at Technion, where we have been also trying to develop systems like this from bottom up. So instead of looking at the cell itself, try to come up with model systems where you can build the systems from what are the minimum components you need to get kind of processes or phenomena that I showed you. And with that, I'm happy to take questions. Equal numbers right and left? Yes, yeah. So the density, yeah. So the plus, I mean, every time you generate a +1, you generate a -1. That's the question. Yes, they do. Yeah. And I've actually been showing you projection on like kind of like so this happens on a surface like a sphere. And I've been showing you the projection, but now we actually have the capability to image the whole thing and kind of look at questions like that.
Thank you for the talk. Two quick questions. So first, is there a way that you think about extracting whether there's one fundamental time scale to the energy injection versus multiple?
Yes, I love that question. So, yes, yeah.
And second really quick question is, why don't you just correlate the probe positions of these point source spheres in space and build the space-dependent correlation function as opposed to using, why do you need to use the carbon nanotubes at all?
Okay. Okay, so the first question is that is something that, so we've been pushing that in terms of theory. So in the theory, in the simple model that I showed you, you can indeed introduce different active noise with different time scales. And what we see, and I don't have a slide on that, if you have enough separation of time scale, you can actually observe different peaks. So that I think, so theoretically, that's possible. Experimentally, I'm really hoping that we would be able, again, if you have enough separation of time scale, I would hope that we would be able to kind of see that. And for the second question, so it's certainly, actually, KL divergence is really like kind of looking at an endpoint correlation function, right? And it's certainly, so we've also been trying to do that as well. In particular, carbon nanotubes is interesting because it's basically across a single kind of one probe. You're looking at many different scales. This is with the probe particle. Obviously, that's also possible. And probe particle, actually, it's the spherical probe particle is even maybe a little easier. Nanotubes are fancier, so. But there's also interesting physics there. But the probe particles, and that's actually something that we are interested because, like for instance, this spiral waves that I showed you. Indeed, the spiral waves are happening in the same system where we have the spherical probe particles, and now we're kind of like trying to correlate like this. Again, their dynamics or the irreversibility or whatever metric that we measure with kind of like the properties that we can measure from the spiral waves. And yeah.
Sometimes this game, I know people want to propose principles of entropy or entropy production. You can either minimize or maximize depending on constraints. Do those kinds of things, is that an interesting way of looking at your results?
I think that's a very interesting question. So, like when I first saw the peak, like I was like, okay, maybe this is kind of like an idea of like you're really kind of maximizing the entropy there. I would say that I would like to answer that question maybe kind of looking at more different systems and then being more confident about this actually is indeed the case. But it's definitely something that we are looking at. And this ideas of, I talked about energetic landscape. To me, when you talk to biologists, there's always this questions of, accuracy, fidelity. They need more. You need to dissipate more to get more out of your system. And I think I'm really hoping to maybe kind of like relate these or what we see to questions like that. And that's certainly.
Anything else? Okay, well, thanks, Nikta, again. Cool. That's lots of fun.
Okay, thank you.
I'm going to see if actually Rachel